All right, let's start our lecture on meiosis. And as I did last time, it's going to feel a lot faster, but that's because I'm expecting you guys to stop, take notes, talk to each other if you're doing this in class, see if you have some questions that go along, and we'll just keep moving forward. So just wanted to start out with the chromosome number. You have homologous chromosomes. Those are chromosomes that are the same number, essentially. You know how we said that we have 23 sets of chromosomes? The ones that have, the first chromosome has the same genes and the same sets, and the second would have the same too. So homologous chromosomes are not identical chromosomes because they might have different genes on them, but they are the same chromosome. One comes from the father and the mother of the individual. So when you are diploid, you have both sets of chromosomes and you're called 2N. And when you are haploid, you only have a single set of chromosomes and you are 1N. So most of all the conversation we've been having up to this point has been about diploid cells. Those are body cells or somatic cells. We are now going to start talking about haploid cells a little bit more often, which are the gametes. These gametes are the ones that we represent in Punnett squares when we are showing the ones that we're showing in the Punnett square. Red dot. So this was, if you guys, I might make this so you can do this individually or at the same time as we are doing this, but I'd like you to go ahead and drag the red dot onto the spot where you have 2 full copies of DNA or a diploid cell, and an orange where one would have half. So take a second, pause, look at this, and you guys should be able to go through that. So here we have the genetic information, and we have G1, S, G2, and the mitotic phase. And we've already talked about that, except this has been the one that we've talked about, right? And we've talked about how there's checkpoints in G1 and G2 and the M phase to make sure that the cells are going appropriately. We've also said that 90% of the time is spent in interphase, which is this G1, S, and G2 phase. And we have this small mitotic phase that exists down here. And this S phase is where genetic material duplicates. I'm just trying to remind you of what we've learned, which happens in mitosis. In mitosis, we have prophase, metaphase, anaphase, and telophase. PMAT. Sometimes you guys can have different mnemonic devices that you can do. That's up to you. If you decide to come up with one, I think PMAT, prophase, metaphase, anaphase, telophase are all pretty easy, and we've discussed a variety of ways to kind of memorize what's happening there. Now we are talking about meiosis, and if you guys can take a small bit of time in your self-guided pair deck to go ahead and try answering this. And so if you see in meiosis, we have 2 sets of rotations, and I want you guys to think about what kind of cells would need to be haploid and wide. So if you could type your answer in, I'll scroll through these later. You guys can discuss it afterwards. I know that you guys are going to be doing this whole class probably like last time. But take your time, pause the video, and write a small response. All right. So we are on to prophase 1. And prophase 1 is the place where in meiosis, so now we're in meiosis, and as you guys may have answered here, gametes or sex cells are going to be the ones that do meiosis. And so that means that we aren't trying to make cells that have completely identical cells, right? We need to have 1, half the genetic information, and 2, because one of the great advantages of sexual reproduction is genetic variation, we want to add especially more genetic variation. So here we have prophase 1. And in prophase 1, once again, the nuclear envelope has dissolved, and you can actually see your duplicated chromosomes because they have formed into the chromosomes, right? And they are formed in the center by a centromere, and we have the centrioles forming and the spindle forming, and the nuclear envelope is dissolved, right? So what we have in this point is crossing over. And if you guys just, take a second to try answering in your notebook, not in your notebook, in the pair deck, what's going on here, and what happens, right, is we have these tetrads. They go over each other, and then they come out with, if you notice, these are color-coded, one set of chromosomes that is no longer identical to the other. Remember right here, these are copies of each other. During the S phase, they are copied, and this is identical to this. After crossing over, we have a situation where no longer are these sister chromatids, they've been mixed up. And so if you guys take a second to answer what's going on there, hopefully you guys discussed that it actually adds to genetic variation, because no longer do you have identical chromosomes, but you have an opportunity to send one of 4 genetically unique chromosomes to the next cell. And here we are in metaphase 1. Our chromosomes that have finished being crossing over are going to line up in the middle, but as you notice, they aren't lining up in the middle in the same sort of way that you would before, right? The chromosomes are lining up, but now we have not identical chromosomes. We have completely unique chromosomes that have been matched up, and instead of being pulled apart, the entire homologous pairs of chromosomes will get kind of moved. Now, once again, I'd like you guys to take a small second and tell me in your pair deck, what does the X shape indicate to you? All right, and remember, when you are in the X shape, that means you've been duplicated, so we have twice the amount of genetic information. Here we have anaphase, and they no longer make that little A, but you can see during anaphase, they are getting pulled to their either part, but once again, they are getting pulled at their centromeres. The X's aren't separating, and it doesn't matter that you have completely identical one set to each side, because we want genetically unique cells, right? And why would you pull homologous chromosomes to either side? I'd like you guys to think about that. Take a time to answer. And remember, the reason we'd want homologous chromosomes to each side is we still need to have the same chromosome numbers in each one. They just don't need to be identical because we're trying to make a half cell. And then during this telophase in cytokinesis 1, we now have, once again, cells getting split up with their homologous chromosomes in each side. Homologous meaning that they're set up, but they're still in that X form, which means that they've kind of been duplicated, right? So we have still the genetic information that we need in each one. So are these cells identical? Will they be haploid or diploid? So if you guys could go ahead and take the chance to answer that. And I'll kind of just wait for a second, pause the video here. And I would say that these cells are not identical. And we already know that because we have some crossing over that made them. Are they going to be haploid or diploid? They have the complete genetic information they need, so they'd be diploid, right? And sometimes I remember there was a student earlier last class who called this cell right here a double diploid cell because it does have twice the genetic information that you would need. So now we're prophase 2. So what we are doing is we're starting the cycle over again. This cell, has divided. And it just happened. And now we are starting meiosis 2. So meiosis is basically 2 consecutive cell divisions. The first one happens with crossing over, and the second one happens here that we're about to start. So prophase 1, we have our cells that are set up. We have them that have been crossed over, so they're now genetically identical. And then you kind of have to start asking yourself, how many chromosomes and how many chromatids do we have, right? And, have a short conversation to see if you can kind of figure out how many. All right, so now we are seeing what we've seen in the past, right? Our chromatids are going to get split up from our chromosomes. And right, this was once from mom, this one once from dad, this was once from mom, this was once from dad, and in each of them, they are now starting to get pulled apart. So while it wasn't the same genetic information because they were still crossed over, they weren't identical, but now we're pulling them apart, and we are going to have one of 2 chromosomes, right? The chromatids in each, right? So remember, if you have one of 2, 1 out of 2 is 50%. So these phases are going in to create a cell that has 50% of the genetic information that is genetically unique. So now what we have done is created 4 cells that have and because I'm doing this like this, I can't, magically make the words appear the way I'm presenting this. But in this one, the end product of meiosis is that you're going to have 4 daughter cells that have half the normal chromosome number. And not only that, but they are all genetically unique, which has really increased the genetic variation, which is really important for, see if you guys can answer it, biodiversity, right? Genetic diversity adds for biodiversity, which allows for evolution, which allows for survival of the fittest and changes to happen. So once again, here's our summary of meiosis, with the cell first getting some crossing over, splitting it up so we have chromosomes in each set, then each of those chromosomes split so that we have 50% of genetic information that have 4 genetically unique daughter cells. A comparison between the two, right? In both of them, we're starting out with parent cells that are 2N, then we get the double diploid, right? In this case, the tetrads align at the metaphase, where these ones, chromosomes align in the metaphase plate, right? But in this case, the tetrads are aligning so that we are actually splitting them not in half like we do here, but so that you're getting one of 2 of the chromosomes that have been crossed over. Then we separate them, they split again, to pull apart to create 4 unique daughter cells. So this is something that you might just need to make flashcards on, try to remember the purpose of, read over and over again. The value of sexual reproduction. So once again, the reason why sexual reproduction is valuable in terms of the sense of evolution is that it's the driver of natural selection. You have the independent assortment of chromosomes that they line up completely randomly in metaphase 1, right? So if we look back to right here, you can see this random alignment of the chromosomes during metaphase 1, or is it right here, means that you might be getting moms, you might be getting dads, you might be getting a mix of the two, and that lining up is going to mean that there's no guarantee of which combination of chromosomes are going to get passed on to your sex cells. Crossing over adds the genetic information, and then random fertilization, which means that you never know which sperm is going to fertilize which egg. And as a result, you can have all these combinations, and they show them here, of chromosomes, which can lead to all these different phenotypes. And this is why genetics is much more complicated than what we started out with Mendel. So if you guys could go ahead and write a response, we'll kind of take a little pause here. We have sperm plus egg, and you can see that any 2 parents will produce a zygote with over 70 trillion, and I can't fathom 70 trillion, and I'm not even sure if you can fathom 70 trillion. If you had, I bet there aren't 70 trillion grains of sand in the world. Like 70 trillion is such a huge number. I'm positive that there aren't. I mean, there might be 70 trillion atoms in like a city or something. It is huge. So if you guys could please take the time to answer why variation is so important, and then have a small discussion about that when you write it in your pair deck. And then we'll move forward to the last bit of the slides. Gene linkage. So remember how we talked about how cells are randomly, how there is the law of independent assortment that came with Mendel, which means that your chance of getting, especially in a dihybrid cross, your chance of getting a pink flower is independent of the chance of having a wrinkled seed, right? That those 2 genetic ones are completely random. Well, it turns out that's not always the case, and that makes sense, and Mendel didn't know this at the time, but if your genes are located near each other on a chromosome, that means that they're going to be linked. So, can you please take a little bit of time to write about this, and then we can have a small discussion about how these differences can happen. And then gene maps. This is going to be, we're going to on Friday do a Drosophila activity. And so what we're actually going to do is we're going to be able to look at the flies like we have to kind of get an idea of what phenotypes they have. And then the cool thing about flies is they only have, I want to say 2 chromosomes. So you can actually know exactly where each one of these genes are on which of the chromosomes, which makes it very good to be able to study. So I think a small short discussion, and hopefully this didn't take too long, but hopefully it took longer than 15 minutes as you guys did this on your own.